So cool.
Yeah.
I can't survive.
I don't get here's why. Well, it's especially with if I didn't teach it, it's a great piece of I love that you were raised big like around like came out this is probably where they were using I'd like to figure out how to make that. Thank you so much.
That sounds really interesting.
It is.
Oh, cool. Thank you for including that. Because last week I got marked off-ish. Oh, I saw you first.
Really?
Yeah.
That's awesome.
So you don't, I just don't order from anybody
from
who
us.
has exactly what I'm looking for.
No, that's
All right, folks, we're gonna get started. Real quick, I passed out your papers,
Dr. Robertson, do you want to click it?
and
No.
I have
Just checking.
added an extra form that might be helpful. So
I don't know.
you guys don't have to do citations for the research papers we do at the end, like in a literature cited, but I do want you to cite them into text correctly, as you would if you were maybe writing a paper. So I picked a format that's specific to usually wildlife journals and a lot of life science journals that I want you to use. Recognize that the journal you pick, the subject you're in, these might change, but for this class, this is how you need to do it. So if you're referencing one author, two authors, or three, or more than two, it's different. So if you're referencing one, you just need to say, obviously, one person's last name. You don't need their initials or the page numbers or anything like that. There are two ways that you can do it. They're both equally appropriate. So if you're just referencing at the top, like so-and-so found this, you put their name, the year in parentheses, so he's indicating which paper you're referencing, and then your statement, okay? If you're just making a statement, like statement here, children prefer M&Ms to Skittles, that's a statement, then you need to back up where you found that source by putting in parentheses, again, with the name and the year. Is that clear? So the way it works is I have references for one author, two authors, you list both names, Anderson and White would be an example, but the rest of the structure is the same, and then if it's more than two, you say Anderson et al. And then the year. You don't have to list all 10 people. Also, when you're referencing papers in your summaries, you don't reference the title of the paper. You just say, Robertson 2010 studied this and it was awesome, or whatever. You don't have to put in all that other stuff. Yeah, and that makes it succinct. Then we know what paper you're talking about. You don't need to talk about their first name or any of that stuff. Is that clear? Okay, great. Your reflections last week were great. And the new reflections are going to pop up after class, so that's all set. So with that, I'm going to introduce our speaker for today. I have to tell a little personal story about Ian. He gave me my very first job in biology when I was, I think, a sophomore. I showed up in his lab and talked to his graduate student, him, and said, I don't know anything about insects or plants, and I've never done field work, but I'm ready. Hire me. And they did. So that's good for you guys to think about if you don't have experience. You can always go out and try, and probably somebody will hire you. So hopefully he doesn't regret it. He has been at the university for, gosh, 15 years?
16.
16 years. He's been here for a while.
Since 2000.
We haven't aged at all. We look the same as we did when we started 15 years ago.
The picture of my office door, and he'll change your mind.
And yeah, with that, I'm going to let him sort of go over his introduction and topic. And
All right. So the reason I hired him was that in Biology 192, I was the instructor, and she came up and I don't know if it was frequently or at least on one or two occasions asked some questions that were just really good questions that are not typical. Usually you get the will it be on the exam or stuff like that, and it's like, oh, whatever. But they were good questions and probing questions, and it's pretty good. So it sort of got me thinking that way, and the lack of knowledge doesn't matter that much because you don't expect a lot of experience that way. You want someone who's interested and hardworking in the field. So that all works out. So, seed predation, we're going to talk about. Before I do, I thought I'd just try and broaden this out a little bit by showing that. Clearly not seed predation, but I did want to ask the question, what is predation? What do you think predation is? Blurred out. That's predation, or soon will be.
I thought it was when one animal ate another animal.
So one animal eats another animal. Okay. Anyone else have a?
When like a predator would inhibit the recruitment ability of the prey.
By recruitment ability, you mean the?
Like the ability to reproduce effectively.
Perhaps. Let's see if we can narrow this down. So I guess what I want to get at is I call these things seed predators. Clearly it's not an animal eating another animal. And so is it just a case of it kind of sounds cool to call them seed predators? And so that's why we use it as sort of a jargony term. And I don't mean it exclusively as a joke because my Dr. Beltoff in our department right next door, one day said, why are you calling them predators? He works on raptors and owls eating things, and those are predators, we would agree. It's like they're eating seeds. So the question is, what is a predator? And you were sort of getting at it. If it's not predation, then what we call it herbivory, because it's eating a plant, right? So we'd say an herbivore. So then I could ask, what's an herbivore? What are they doing? When you think of something that's an herbivore, what's it doing? Eating a plant, right?
It's eating a piece of it.
Eating a piece of a plant. And so what does it do to the plant?
Reduces its ability to function.
So it's a negative, right? It's declining. When an animal eats a seed, what is it doing? We kind of do this in psychology.
Basically, the seed is the child, so it's eating the child. So the child, it's predation because it's
eliminating
killing the living
an entire organism, right? Which is sort of it inhibits the recruitment. But so does herbivory because it could lower the success of a tree, and so it doesn't have as great reproductive success. But with herbivory, you're getting this sort of the plant is being consumed, but it's just portions of the plant being consumed. Typically, the roots are still present, even with grass being snipped, there's still roots, and it grows back up again, right? So it's not being eliminated. Whereas a seed is kind of like the zebra. It's getting eliminated from existence, and it has no future. So if herbivory is a sort of gradual consumption of plant material, then I would ask, what do you call it? What is a parasite? We talked about this in insectology, so they're sort of nodding.
I like that.
What's parasitism?
An organism living off of another organism and decreasing its fitness.
Okay.
Same thing as a herbivory.
So what is it? And the reason I'm sort of drawing this through is that there are some who would argue that herbivory, in any sort of ecological sense, is really like parasitism, just on a macro scale. It's not microorganisms in you, but when you think of caterpillars eating the leaf of a tree, and it could be very similar to having parasites that reduce your overall sort of physical well-being or fitness, but not your survival. Yep.
So I'm just.
That's all right.
are we saying like for parasitism, it's, you know, it's just taking and not giving, that's giving back, that's like the definition of
Yep.
parasite.
Right.
Could it really be the same with the vivary when they're creating somewhat of fertilizer out of that plant material for that plant material?
So you think the droppings of the caterpillar?
So could that be like, I don't know, I'm just.
Yeah, no, that's I hadn't thought about that. I guess you would sort of look at it in terms of net cost and benefit. Parasites tend to debilitate their hosts, and herbivores like a caterpillar, even if there's some, you know, their droppings might fertilize, the loss of leaf tissue is going to, and the reduction in the ability to photosynthesize and produce resources, you know, it's probably going to have a net negative effect. So in many ways, herbivory and parasitism are sort of aligned, at least in a theoretical sense. And whereas predation, it's not debilitating an organism, it is killing and consuming it. And seeds are being eaten and removed from the system. And so it's not a stretch to call that predation, even though it's not an animal situation. So I thought I'd just throw that in there at the beginning, just so we think about the terms. The reason I bring it up too is that in writing lectures a couple of years ago, I got to a section on herbivory, and this ecologist went off on a big tangent about parasitism. I'm like, well, I wanted to define herbivory, not parasitism. And then they sort of brought it full circle and said it's kind of the same thing. So I'm not going to talk about that. We're going to talk about ants and plants in Idaho. And this is a study system I've worked on for as long as I've been here. Well, no, the plant is. The ant's more recent. But this is what we're looking at. This is a harvester ant colony. There's only one species of harvester ant here. It's called Pogonomyrmex salinus, the Hawaii harvester ant. It's the northernmost species of Pogonomyrmex. The genus is found throughout the Americas, South America, North America, Central America. And they're very common in arid and semi-arid habitats. And what you see here is it's sort of, well, now it's more and more its natural habitat, even though it's highly disturbed habitat. And for the ant, which is an obligate, well, it's primarily a granivore, it eats seeds. And this is like it's living in an ocean of food. There's all these different types of plants out there. These are various seed-bearing plants that the harvester ants will eat. There are others, but in the system I'm working with, those are the most common. This is tumble mustard, that's an invasive weedy species. Poa secunda is a native grass, sandberg's bluegrass. And Bromus nectarum is cheatgrass, which is an invasive species. And then Lepidium papiliferum is sort of the focal plant that I've been studying for different reasons over the years. And that plant, you can see it right up there on the top, is a rather unique plant in that it's endemic to southwestern Idaho, which means it's found nowhere else in the world. Its range is from about the Oregon border to Glens Ferry along the Snake River Plain, and then there's another population a little further south. That's the only place in the world it's found, and it has very specific habitat requirements. So it's something that has been a source of funding for me because local agencies are interested in its conservation. There is a range map. So these are that's the Snake River Plain populations, or some of the foothills here, and then there's a disjunct region, and it's called the Jarbidge area. In the yellow shows the overall range, but I'd want to point out that it's actually isolated little pockets where it's found. There's many areas where it's disappeared from because of agriculture, urbanization, habitat loss of any form. I mean, it really is getting hammered that way. It has very specific habitat requirements, which I'll bring up in the next slide. Something that will be of interest, perhaps later in discussion, is that it's really become a center of controversy because of conflicting interests in terms of habitat management. So there are those very interested in the conservation of this rare plant, but there are land use issues, specifically cattle grazing is a big industry in this area. And so there's a lot of friction between those competing interests. And, you know, since I've started working on it, it's been proposed for listing under the Endangered Species Act 3 times, I believe. The first time, it was in the early 2000s, it was denied listing based on, I don't know, the Fish and Wildlife Service delivers a ruling, and the reasoning they used for it seemed a bit odd to me. It was challenged in court. Western Watersheds took it to court, and it was overturned by a judge. He stated that the reasoning was spurious or capricious or some weird. He did not like the way it was argued, and so they threw that ruling out. It was then re-proposed for listing, and it was eventually listed as a threatened species in 2009. And then they were sued by other interests, and that was overturned, saying that the data were not sufficient to demonstrate the plant was in peril, that it was at risk of extinction. And so that left it with no status. Fish and Wildlife Service now is again reviewing, or is now putting up as a proposed listing. No decision of whether to list or not has been made, but they're going through that process again. And so in 15 years, this is 3 times, and you can bet, whatever the decision, there's going to be somebody suing over that decision. I mean, it's just, there's a lot at stake here in terms of, well, I don't know if it's bragging rights, but just there's competing interests, and no one's going to be happy, or one group is going to be unhappy, the other happy. So that has created an interesting environment for me to work in, and perhaps later we can talk about that, because it got me in a little bit of trouble here. Not trouble, but it caused issues for me to deal with, because some people in the public aren't happy with what I do, and they complain about it. So we can talk about the importance of tenure and research. But I've been viewing this as my role, I think, is that research can help inform decisions that are made. It can provide data that can be used in these decisions. And what I would like to suggest is that I don't feel I'm an advocate one way or the other for this plan. It's not my job to be an advocate. It's my job to conduct research and say, here's what I found, here's what we know, and then resource managers can apply this information. Now, in terms of conservation management strategies, it doesn't mean I can't have an opinion about that, but the data drives the opinion. I don't already have an idea of what has to happen. I look and ask questions, and then just present what I found and tell them what it means within the bounds of what the answers can tell us. So this is some of the nice habitat where the plant is found. This is slickspot peppergrass here. These are larger plants than in the previous slide. And I show this slide because it's a little more representative of how this area used to look throughout much of the region. Sagebrush forest, and with very low understory, not a lot of grasses in here, it's fairly sparse. And then slickspot peppergrass lives in microsites known as slick spots, which are unique geological formations or depressions, slight depressions that have a hard pan layer on the surface, and that creates sort of a boundary that allows water to be held in clay down beneath. And so the moisture levels are higher in these areas, but these areas are also much higher in salts, dissolved salts, and that is pretty inhospitable to most plants. And so these things, which are called slick spots, tend to be fairly bare, but then you have this one plant, slickspot peppergrass, that specializes living in there. And so you see them sort of growing in these patches, and the sagebrush is all around the perimeter. You have, in this picture, you have things like burr buttercup, which is an invasive species invading into the slick spots, but for the most part, it's fairly open. The plant pretty much lives only in these slick spots, and they're very patchy in the environment. When it flowers, the insects pollinate those flowers. The fruits produce seeds. The seeds drop to the ground, and they do not necessarily germinate the next year. The seeds can remain what's known as a seed bank. Our estimates right now are for like a decade they can still be viable. It might even be much longer than that. That's a really important aspect of desert biology or seed banks for plants because it allows them to weather the bad years and still have persistence.
So with the slick spot peppergrass, is it just exactly where they are growing that is considered a slick spot, or in the case of this picture, is it that entire circle essentially?
Well, in fact, this is more of what I would call the slick spot. They look a little different depending on where you are. Some of them are very well defined, where it's very smooth hardpan, and then a margin with grasses around it, and you can just see the patch. Here you have things like burr buttercup, which have been invading into what used to be probably a more pristine slick spot. So it's not quite as well defined as it might be. And I'll actually talk a little bit later about why you're seeing them perhaps at the margins. We're trying to figure that out. So the seed bank is perhaps an important issue there. Plant numbers vary year to year. It tracks a little bit with, or a lot with precipitation. So overwinter rainfall is important. But what we're finding out is it's even the timing of rainfall is critical, because if it happens at the wrong time of year, it can drown the plants. At other times of the year, it's perfect for setting them up for the next season. And so when I go out in the field, I always feel kind of surprised, or I never know what to expect. Will it be a good year or a bad year? A few years ago, I had a grad student go out and we almost couldn't find the plant. It was terrible. It was our second field season, had all these big plans, and a site that sometimes would have 10, 20,000 plants had 19. And that was what she was dealt. That was just a terrible experience for her and me. Okay. So on to what I'm going to do is present basically the work of several graduate students, showing you sort of the evolution of this study system for us. You know, answering some basic questions, and then they lead to new questions, and then I'll finish up with how we're sort of trying to apply this information in rehabilitation for the species. So it began actually with one of the papers you read. Just a simple observation of seed predation. And it came about, I had been studying the plant for years and really had no idea the harvester ants had anything to do with it. I had been studying pollination. That's why I was first contacted by the Bureau of Land Management back in 2001 saying, they asked, hey, could you figure out how this plant is pollinated? They've been monitoring it for years and had no idea how it even reproduced. It's a funny thing for me that a lot of the agencies are very good at sort of counting stuff year by year, but they don't know anything about it. And so they asked me to figure out how it was pollinated, so we did some work that way. And when Josh wanted to join my lab, I said, well, why don't you go out, spend some time out in the field, and look around and sort of just see if something captures your attention. And he went to one site out in Eagle, and he said, there are ants all over these plants, and they were taking the seeds away. And that was totally new to me. And it was because I've been working on pollination, which occurs earlier in the spring, or in sort of May, June. And then when that was done, I kind of wrapped things up and just sort of said, oh, we've done our study. And the site Josh went to, turns out that site's always about a month earlier than the other sites we go to. And so when he went out there, it had long finished its flowering, and it was in seed, and then he was seeing all this activity. And it was just, we had never really paid attention to it because we had finished up what we were sort of tasked with doing. So it was a good lesson of, learn about your study organism, really sort of, think beyond just the immediate thing you're looking for. And it turned out important, or some of the things I missed was, years later, I have no idea if harvester ants were really that abundant at the times when I was doing the pollination work. I didn't even think to look because it just wasn't what I was looking at. And it would have been very helpful if I had. Anyway, here's what he was seeing. Ants crawling up on plants. These are the harvester ants crawling onto these plants and actually clipping fruits. This is a fruit, and inside it there's two seeds, one in each half there. They were clipping them and taking them away. And we also saw them taking the seeds that had dropped to the ground. That's the actual seed. We saw them taking it off the ground. And so the question Josh asked was simply, how much is really being taken? They're eating it, okay, but what does this mean? So this is the study he did, where he simply, he got paired system here One group of the pear was exposed to ants, the other one was shielded from ants, and we just looked over time what happened to the seeds, and we found that in the ones that were shielded, the percent of seeds lost or fruits lost remained near zero. They weren't being taken by anything. They had them blocked off. And over time, the plants that were exposed to ants kept losing their fruits, and we just saw them keep disappearing. We had to stop when we got to about 7 weeks into the study because these fresh green fruits they were clipping off. You can see how it's been clipped here. It's just that's what they were doing, and we could see that real obviously. As the fruits matured, the casing dried, and instead of clipping the fruits off, the ants would just grab it and twist it, and it would split, and then they'd pull the seeds out and walk away. And we couldn't tell anymore whether they were taking seeds or the seeds were just breaking off and dropping naturally. So we had to stop it here. We observed, ongoing, we observed ants still climbing on the plant and grabbing stuff. We just couldn't document it as well.
Did you know that going in? I mean, I'm
No.
assuming you didn't. Like, how did you know? Like, oh crap, 7 weeks in, like we have to stop.
Well, it's kind of how he's figuring that out when he's watching it. We're just seeing what's happening. You know, you'll learn as you go. So we were getting fairly high levels of loss here, but we knew it was going higher. He also did an experiment where he put seeds on the ground, and we found within 3 hours they were just almost all taken. I mean, we were putting out 10 seeds at a time.
So, basic question about ants. Do they work 9 to 5? Like, what is is this around the day, sort of 24-hour ants are going and
That's a good question.
preying on these seeds, or?
No, it's not. They are, around here, they're active bimodally. They're sensitive, so during the night they don't forage. At least this species doesn't here. In the morning, as it warms up, they become active around 8:30 in the morning. And then they remain active foraging till about 11 or noon. Sometimes in the afternoon, depends how hot it is. When the temperatures start to get really hot, midsummer, they just totally close up. And then in the evening, they'll come out again when it cools off a little bit.
So you can target your research.
Yep. So we always did mornings. We didn't go backwards in the afternoon or in the evening.
What's the numbers in parentheses mean about week?
Oh, sample sizes. So we had 12 pairs initially, but the problem was that as we got as the season progressed, we were starting to get to a point where we couldn't tell anymore this number because of that problem I was saying. So we had to basically ditch some of our pairs for further analysis. And once we got to just 2, it was like, there's no point in doing this. Yeah, thanks for asking.
Can you talk just a little bit about the ant shield mechanism? Like, is it just plastic that was keeping them away from it, or?
Yep, it's this stuff here. It's garden edging, actually. You can buy it at Home Depot. We just wrap it around and seal it into a circle. And then we stake it to the ground, so it's flush with the ground. Now, because of the way this is, it has a bit of a lip here.
Mm-hmm.
If ants were climbing up it, they get to that lip and they just couldn't navigate over it. And so it stopped them from getting in there. You'll find when you read papers and the methods they describe how they do stuff, it always sounds very scientific and professional. Half the time it's margarine containers, milk cartons, garden edging, stuff like that. It never looks as good in the field. When you take the photo, that's why you don't put a lot of photos in your papers. People would see that stupid. Because all the molecular biologists have their flasks and coats, and it looks so scientific, and we have that. It still works. So, to give you a sense of what this really looks like on the ground, because this is, fine data on a sheet, but here's what a plant looks like when it's fully fruited. So it's drying out, lots of fruits there. Eventually those husks or the fruit casings will split, seeds will drop to the ground around the base of the plant. Here's one that's been exposed to ants, and you can see that every stem has the base of the stem where the fruit would attach is there, but there's a couple fruits right here. The rest of the plant has been skeletonized. There's just nothing left. And just to show you the magnitude of how much can be accounted for by this predator. And when you put it in the context of a rare plant, that's going to send some alarm signals. These are not seed dispersers. They're not just carrying them off and dropping them they are granivores. They eat the seeds. They take them below ground and they eat them. So they aren't benefiting the plant. And as was in that paper, Josh followed 100 ants and showed that they always delivered their fruit to the nest. He was crawling on the ground. Again, a little lack of dignity in there, but okay. So that's at a sort of plant scale. I thought I'd show you what a site looks like. This is one of my main field sites. It's Kuna Butte. Here's a scale, 100 meters here to here. And what you're seeing, the triangles represent ant colonies at that site. We surveyed them, GPS them. And the green dots represent patches that had lepidium growing in them. This is a good year. There's lots of lepidium. The red triangles are ant colonies where we found evidence that they've been eating seeds from lepidium. And you can see how it's all sort of centered right where the plants are. And so all those ant colonies were consuming lepidium seeds. So there's a lot of exposure, but it's not always like that. Sites like I showed you earlier, that was that scene from earlier, where there's sagebrush and low understory, are very different than that one, which is a disturbed site that has lots of cheatgrass, lots of open areas that burned back in the late 1990s. Sagebrush has been removed. You can see just about there, you can see a skeleton of an old sagebrush. They don't return very easily. So I did some work looking at habitat associations between harvester ants and their environment, and where were the ants most likely to be found. And this is what we came up with. I'll explain. The vertical axis here is ant colony density, so the number of colonies per hectare. And so bigger numbers mean more dense, like this. Okay, so up to 60 colonies per hectare, which is just really dense. 15, 20 paces, you'll hit another one when it's that dense. Down here, we have sagebrush cover. So no sagebrush, lots of sagebrush. And here, I have grasses. In fact, I have non-bromous grasses, and I'll explain that in a minute, from 0 to 10% cover. Lots of areas out there are just bare ground. So what this tells us is that when there is not much sagebrush and lots of grass, non-bromous grass, you have the highest densities. That's like this. No sagebrush, lots of grasses. Okay. When you have high sagebrush cover, you don't get very many ant colonies from it. They don't like overstory. They like open areas to nest. Now, this is really an important result because if you think of what's happening to the environment out here, we are losing sagebrush habitat through fire. We have cheatgrass moving into areas. Cheatgrass burns, so it's changing the fire cycle in this area. So it used to be about every 50 or 60 years you got a fire. Now it's about every 5 years you get a fire in any given area. And the sagebrush gets knocked out, and before sagebrush can ever establish, cheatgrass is right back in there or in there for the first time, and it's just totally changed the habitat. If you drive down I-84 between here and Mountain Home, and you just see open areas, that's not how it used to look like. And it's creating ideal habitat for ant colonies. And we know ant colonies separate the seeds of this rare plant. So that provides sort of the ecological context of what we're dealing with. I had another student come by after Josh did his, and he was looking more specifically at what are the ants eating. We know they eat lepidium, but what are they eating in this sort of greater matrix of food availability? He had that question, is what type of seeds do they harvest? We know they eat this stuff, but these are other prominent species. Bluegrass is a native, cheatgrass non-native, tumble mustard, another mustard like peppergrass, but a non-native mustard. And well, you can tell they're eating them because I've showed you pictures of them all with the seeds in their mouths. So there they are there. Cheatgrass has really big seeds with long spiky awns. That's going to come out as important. Sandberg's bluegrass, a small seed that they can easily tuck in their mandibles. That's pretty big, but often they just, after Josh's work where they're taking these fruits, That's not how they usually do it. They usually take the seeds once they drop to the ground. It was really kind of a special case that they were clipping fruits off of plants. We only found that out in subsequent years though. And then tumble mustard, there's a seed there. It's even tucked one under its chin. So it's carrying multiple seeds at a time. Chin is the technical entomological term for it. Okay, so that, we know they're eating those things. But Josh's question, or Matt's question was a little more specific. He wanted to know, are the seeds being consumed in proportion to their availability? So are ants simply responding to what's out there and just, if there's doubling on of that type of seed out there, they'll take double that amount in their diet. So is it proportional, or are there certain seed types that they prefer over others? Simple question. Here's what he had to do to figure that out. On a weekly basis, he had to figure out what seeds were available to the ants. So he did a bunch of core samples around ant colonies. He'd take a bunch of samples like this. He'd pull out a core, he'd dust off all the seeds that were sitting on the surface into a petri dish, and then take a whole bunch of samples, random samples within the foraging range of an ant, put them together, and later on have to go through that and figure out all the different seeds that were available. That worked for certainly cheatgrass and bluegrass, not for this. And I'll talk later about how we did that. It worked okay for this, but probably underrepresented. So what seeds are available, and then he spent time figuring out what are they actually eating. He only did this once. There's the ant colony there. You can't see what they're doing. So he was really actually on his hands and knees looking this far away, and he followed ants into the nest for a 5-minute period and recorded what each one was bringing in visually, because they look different. So we could tell that. So he did that over the course of the summer, each week measuring what was out there, and then what they were taking at the time.
Did his observation mess with their regular routine? Like they see the shadow drop and stuff and run?
No,
Or did
they could care less what you're doing. If you get too close, they bite and sting you, and they're pretty nasty. So they're not really worried about us. Or so you're right though. That's something you should be aware of. When we did the pollinator stuff, we had to be much more aware of that because they seemed a little more hesitant when we were around. So we had to sit back about a meter, and we would wait a few minutes before we would even start doing observations to give them a chance to sort of adjust. Okay, I'll give you a sample of what this data looks like. Here's data in 2009. The numbers, the brackets here just mean the week, so the first week of June, 2nd, 3rd, 4th, and then July. So those 2 months. And the different bars represent the estimated availability of those seeds within a 20-meter radius of an ant colony. That's about their foraging distance, how far they'll move. And what you'll see from here is that in this year, POA, Sandberg's bluegrass, the clear bars, was very abundant in early June, but then sort of declined. So you see that curve there. Bromus, this is cheatgrass, was available throughout the season in large amounts. Lepidium seed popped up later in the season. It flowers in June, set seed in July and August. Now, those numbers actually represent computer simulations. Because they are located within slick spots, they're not distributed around the ant colony in a big random way. Using core samples would not ever detect them. And we weren't going to core within slick spots because that would be damaging this very sensitive habitat. So instead, we took a full accounting of how many plants were there, and based on the total number of plants and their sizes, we estimated how much seed would be available over the course of a season, and then we used a computer simulation to basically deposit it. You see it's a very regular distribution, in a normal distribution over the time in which we knew it was there. That was how we approached it, as a way of estimating its availability without actually having to destructively sample for it. So there you see what's available, and now here's what the, yes?
There's no tumble mustard.
No, there wasn't very much tumble mustard there. It is there. This was not a good year for it.
Okay.
Tumble mustard is one of those things that some years you go out and the fields are gold, and this year there was hardly any. And that'll actually factor into the analysis here.
Okay, thank you.
So here's what they were eating over that same period. So this is the number of seeds brought in a 5-minute period of each type. And you see the first week, lots of POA. There was lots of POA there. POA is the bluegrass. And they were eating lots of it, but then it declined. kind of like it, just declined in availability. That was intriguing. Also note, black bars, where are they? They do not like cheatgrass. And this is, we're not the only ones to find this. Harvester ants don't like cheatgrass. We did some other, I'm not going to show you the data, we did stuff with how they handle it, how fast they could run when they were carrying it, and they would get hung up on other branches. I mean, it was so big, and they had these awns, and they were getting tangled up. Multiple ants would grab it, they'd be pulling different directions, and they just can't handle it well. Nutritionally, we analyzed the nutrient content. It's pretty good, but it's just difficult to handle, and so we think they just simply don't choose it because it's not very good to work with. That's why in the analysis earlier, I showed you the with that three-dimensional graph, I said non-bromous grasses. When we include cheatgrass in that, the relationship disappeared in terms of predicting what ant colony density. It doesn't factor in because it's not part of their diet in any big way. Okay, so lots of POA here, but then it declined rapidly, and then you see the lepidium, the slickspot peppergrass, actually dominated the food intake. Intake is lower overall, but there's still, that's the main food being brought in. And a little bit of tumble mustard showing up. There wasn't a lot that year, but it's there. So from this, I would say, okay, peppergrass became the major component of the diet as the season progressed. But a problem for this analysis right now is that this food, which we know they like, wasn't as available later on. So it's hard to say they had a preference for one or the other because it wasn't there. How can you assess that if it's not there to begin with? And so luckily, the next year, he did this over 3 years. I'm only going to show you 2 years out. In the next year, the availability of bluegrass was quite different. It was available throughout the entire summer in larger amounts. Don't know why. The climate must have been more favorable for it. Cheatgrass, most of the summer, was quite available, not early on. More tumble mustard showing up than the previous one, and fairly good amounts of lepidium in there as well. So, slightly different scenario for them. Let's see what they were eating. This is really interesting to me. Before lepidium and tumble mustard showed up, bluegrass dominated. They liked it. It was the best stuff out there. They had a choice between bluegrass and cheatgrass. They go with the bluegrass. And the literature shows they love grass seed, just not cheatgrass seed. But even though it was available later on, when this stuff became available, we see a switch. And here, look at, not a lot of tumble mustard in terms of our ability to actually assess it. That was like dominating the diet. It's possible we just weren't really good at capturing it because a lot of them were not in the turf. They were still on the plants, and the ants were going up and taking them off. And our sampling technique with the core didn't really get at that. But the important thing to note is that in the second half of the season, it was the mustards that were dominating the food availability or the food preference, even though the grass was still available. So it suggests that harvester ants, when given the choice, would really like to eat mustard seed, tumble mustard, and peppergrass seed. Those seeds are tiny, easy to handle. Remember the one that had tucked under its chin? You can grab them easy. And with peppergrass, it's not spread out everywhere. It's in these discrete patches with highly concentrated numbers that are bare, that ants can just walk through without having to climb over everything and walk around. The perfect storm for taking seeds. So we think that's really a problem for the plant. All right. I did have a different graph showing you preferences, but I deleted it at the last minute because I think you get the point.
Yeah.
That when the bluegrass is available, they like it until something better comes along and then they switch. And again, energetically, we had trouble explaining it because all these foods look pretty good to eat in terms of protein, water content, and all that stuff. But the mustard seeds are smaller, and so you can run faster while carrying it. You can get them back to your nest.
I have a question. What happens in the winter? Do the ants, do they just stop eating? Do they die?
They hunker down. No, a colony lasts, well, I don't have measurements for this one, but estimates are 20 to 30 years for a colony. Single queen, and then a worker force that rotates. That's a single nest.
So they just store the stuff down there then?
Yep.
Okay.
And they become pretty inactive. As it gets cold, their metabolism's going to slow right down, so they don't have to feed. They're active from like April to October.
Question to them. Between 2009 and 2010, do you guys have population estimates? Because it seems like in 09, it's like, what the heck were they eating in July?
Well, the ant colonies persist. I did a 5-year study. So the graph, the 3-dimensional graph I showed you with habitat associations, that was something I monitored ant colonies for 5 consecutive years, labeling each one with a number tag and following it to see if it lived or died and how many new ones showed up. Colony numbers didn't change a lot. They went up and down a little bit in some sites more than others, but there was no sort of trend that tracked weather or anything like that. And in a bad year, they could just probably reduce their worker force, right? Have fewer individuals, breed less, and then feed off of stores. And then when things get better, they can ramp up again. So they have to live in a very unpredictable environment. So having that flexibility is good. So anyways, again, the point here is that they like the mustards even when that grass is available, which was kind of, in a way, it's, from the conservation perspective of the plant, it's an alarming finding because it means that the mustards are really a target. Okay, switching gears to a current student. An overriding question I've sort of had since we sort of learned all this is, how on earth does this plant survive, given all the seeds we see being taken? How does it persist? And in fact, does it persist? One of my initial concerns was that because of the seed bank that exists, the fact that you have plants growing in any one given year could be a remnant of reproduction that happened a decade ago. And if ants have been moving into the area as habitat changes, right, as the environment becomes more conducive to ants, perhaps we're kind of seeing the ghost of past reproduction, not current recruitment. So that's why I did the 5-year study, was to see if ant colony numbers are really increasing. I didn't find that. So that's good, I guess. I really wish now, when I was doing pollination, that I had paid attention to whether ants were there or not. I don't know. I don't remember. I didn't measure it.
So, kind of in conjunction with that, this plant is rare, but are numbers decreasing?
Overall, yeah.
Okay.
But there's a lot of reasons for it. The habitat is just getting worse.
Right.
But now it's facing climate change, cattle grazing. Yeah.
But in the areas that are, taking out the human
In some areas it does well.
encroachment, yeah, okay.
Yeah.
So it's,
So, the site I showed you earlier, with all those ant colonies, that's one of my best sites to go to for plant numbers. But it's also the site that had 19 one year, 19 plants, but it was a drought year. And so they weren't coming up for other reasons. It wasn't the ants, it was other reasons. And last year it was a great site. There were lots of plants there. But a question we have is the plant able to replenish its seed bank? Or are ants taking so much of that away that it's really reducing how much can be sort of put down there for future years? And so I have a student now who's finishing up, she'll be defending this spring, working on this question. And so this is the overriding question she's sort of poking at from the edges, perhaps. This is a slightly different question, but it relates to something that was asked earlier. A lot of slick spots look like this, where here is your sort of well-defined slick spot, and the lepidium is at the edges. And one of the ideas we had, perhaps, is that along the edges where there's vegetation creeping into it, perhaps that vegetation provides sort of a more complex environment that serves as a refuge for seeds so that ants have less ability to find them. They can kind of hide out in those in the perimeter, whereas anything on the smooth open area is going to be really vulnerable to seed predators. They just pick it off as it lands. So we wanted to ask that question. Michelle also looked at plants could grow better here than in here, and that experiment didn't work out very well. But I've done other experiments that suggest no, it's not the case. In fact, in my experiments, the margins were actually worse in the center. Another question Michelle was asking is something that we sort of had this idea that, so we know ants take a lot of seeds. And perhaps in many years, when this plant doesn't always have great numbers, sometimes it's just an okay year, maybe in those years, ants are really doing a number on them, right? There's not that many seeds, The ants like those seeds, they're taking all of them, and I go out there and measure and go, oh my God, they're taking all the seeds. We got an eco-disaster here. Like in years like this, well, what if every so often we have just a really good winter, lots of rain, and the next year there's just a flush of these plants. Seeds that were stored in the bank germinate, and then you get these billowing pockets of peppergrass. And then in those years, they produce seed that is in such great numbers that ants, even though they're eating a lot of them, simply can't keep up with those numbers, so the seed bank can replenish. And then over the next few years, some years good, some years bad, it's enough to keep the plant going. And then we have a great year, they replenish. And so that variability might be sort of the key to its survival. Rather than sort of, even though this might be the way most years are, the doom and gloom scenario might not be true if once in a while this can happen. So, and that's where that second paper kind of came in, that idea of satiation. Are there times where there's just, where the ants simply cannot keep up with the seed availability, and as a result, the prey escape and can replenish themselves. So first of all, the soil texture. Michelle did an experiment where she went into some slick spots, and using cages, we call them, they're actually just flower pots. One of them, well, we put 6 in a slick spot. One like this is buried flush with the ground, again, so ants can't get inside it. This one, it's hard to see here, but it's raised. There's a piece of mesh at the bottom that raises the bottom up a couple centimeters, so ants could go into that cage and out of the cage. And then we put 200 seeds in each one of those. Now we did it 10 slick spots. We had controls like this where the ants were not allowed access. And then we had 2 different treatments. We had soil type, the edges, what we call complex soil, or coarse soil, where we thought maybe the seeds can escape. And then we had the center, which was smooth and hard, just smooth sand. And then we also did it near and far from the ant colony. So within a meter or so of the colony versus 6 to 10 meters away, to see if maybe over distance the risk declined. That was the idea. So we put 200 seeds in each cage. Each slick spot would have 6 cages. Yep.
How did you acquire your seeds?
Going out in the field and shaking plants and collecting.
Okay, just
Yep. I got a little vial in my office that's like gold for me.
okay.
It's like 250,000 seeds in there.
Oh my goodness.
It's a jar this big. They're tiny seeds. I didn't think it was that much until a guy from the National Guard who I work with came and he goes, "Oh my God, you've got so many seeds." I think he then said, "Can I have them?" But, so 200 seeds, that's sort of like, you know, what fits on your fingertip. We left them out for 3 days to see what would happen. And this is what happened. In the cage here where no ants were allowed, we were able to recover about 150 seeds. That's our ability to recover. So to recover, we'd go in, the cage would scoop up the top centimeter soil, and then Michelle would go under a microscope, like a dissecting or magnifying glass, through grains of sand, finding the seeds, which are not much bigger than grains of sand. It was awful. I feel like a sadist for making videos. So it didn't matter whether it was on the coarse soil or the fine soil, and this is our control. We found this many seeds. When ants were exposed to them, whether they were close or far away or in coarse or fine soil, they took almost all of them. They just found all these seeds. You know, they're really good at finding and liking lupinium seeds, so they were disappearing. So this pattern doesn't seem to be explained by, you know, we were thinking that maybe that would be higher. But in the coarse soil, some would escape. But we're not finding that. And I've done another study where, again, it didn't happen like that. This pattern might simply be a case of, over the winter, the seeds that haven't been found through just dispersal because of water, they move to the edges and then they get trapped. And that's where they end up. It could be just that pattern. It's nothing to do with the ants. Okay, the other experiment. This was bad. The other experiment was awful. I will never make another graduate student do this. We're moving on. Anyway, so the idea here is we have to know how many seeds are available to ants and relate that to how many are taken. Because we were predicting that as the number available increased, the percentage that would be lost to any individual plant would start to decline. There's some assumptions in that, but that's what we were predicting. First thing Michelle had to do was figure out how many seeds are out there.
Oh,
And here's how she did it. She took a little 5 centimeter by 5 centimeter grid, held it over a plant, and then would see how many of those could fit in the entire slicks of all the plants there, how many of those there were, to get total flowering surface area. That's not the worst. That was the easy part. On a subset of plants at a different site, she took this little grid and then clipped all the stems and counted how many seeds that would represent. So she could then translate her flowering surface area to an estimate of how many seeds were out there. Again, this is the easy part. Then she did part of the experiment was to, she picked 20 different ant colonies, and then within that ant colony's foraging range, which included areas where this plant was, she would pick a pair of plants of similar size, one that she would put this cage, you've seen this cage before, this one's flush to the ground so ants can get at it, this one's raised so ants can get at it, and our assumption is that these plants would have the same number of seeds because we tried to roughly make them similar in size. We'd randomly pick which would be the treatment and which is the control. And so the question was, over the course of the season, how many seeds would be left beneath this plant compared to that plant as a function of how many seeds were available in total, okay? That plant can produce a lot of seeds, and that's a lot of surface area, because to figure out how much was left, she had to go in at the end of the season and take the surface soil, put it in a bag, and go back to the lab and analyze it for, count seeds that are left in the soil. And a plant like this could have 3,000 or 4,000 seeds in the soil. And you're talking a bag of soil this big, going through it, it would take many hours to a couple days for each sample. And she spent hundreds of hours doing this. And they're just brutal. This is why I could not make someone do this again. Especially since the result was not very good. So here is what we found. This is the estimated number of seeds available. We expected a negative trend, and that was the relationship we got. The average take was 82% across here. So they're taking a lot of seeds. But even when there were lots of seeds available, they were still taking a lot of seeds. This is a problem. Not for the plant, but for us. Because there is no estimate in the world that would say ants can take that many seeds. That's 8 million seeds estimated in that area. And the highest estimate I could find that an ant colony could consume this season is around 500,000 seeds. So it makes sense at this end, right up in here, that you could have those levels. It just does not seem possible you could consistently. Remember, we're assuming that this is what happened within the entire foraging area, right? All we could do was sample a couple of plants. And so the concern I have is that in picking these plants, we were either picking certain size classes or certain positions that were more vulnerable to the ants, and that we maybe weren't at the margins or at smaller plants that escaped it better. And so if we had more samples here, we didn't have as many samples at this high end because there weren't that many places where there were that many plants. If we had done more sampling, we might start to get numbers down here because it can't be sustained across the entire slick spot based on that number. Now, that number could be wrong too, but we're not going to be off by an order of magnitude. So this is one of those cases where you try hard and it doesn't always work out. And anybody doing ecological studies knows that this is sometimes what happens. I don't know how easy that's going to be to publish. Probably not very easy. We probably have to spin this a different way and sort of talk about overall predation levels and not try to coach it this way because I'm not convinced by the result because I don't think it's possible. An intriguing effect, sort of maybe even helps emphasize what I've just said, is that we did find a very strong relationship between the activity that occurred in the cage and the number of seeds lost. So we know that we are relating properly the activity of ants to how much seed is being taken. That is working out. So while that number can't be sustained, it is explained by the activity of the ants at that plant. And again, it may mean that we're missing plants where the activity was lower, and then there would have been fewer seeds there. Does that make sense?
Mhm.
This was kind of comforting to us in the sense of, at least we aren't totally out to lunch about everything. It means that when ants are more active under a plant, we can predict that it's going to lose more seed. Don't worry about the scales here, that's a statistical issue for distributions and stuff. So, there you have it. That was an issue for us. Okay, so I want to now I only have a few more slides. I'm going to tell you where I want to go from here. And that is looking at this from, and this is something the Fish and Wildlife Service, who I work with, and the Idaho Army National Guard, they're big funders. This is where ultimately we want to go with these things. So I've been providing, I said earlier, how science can inform. Well, true, but now those agencies are also saying, okay, yeah, we're happy to fund your research, but let's do it in ways that are going to give us things we can actually see results from or get to the actual management. So there's several ways that this can be approached. The bigger question for all this, the bigger approach would be this: rehabilitate sites by promoting sagebrush. This is not stuff I'm doing. Make it so that we know that harvester ants don't like to be in areas where there's lots of sagebrush. So one of the big goals here would be get rid of cheatgrass, get sagebrush growing again. Sure, figure out how to do that. There are people that want to do it. It's not easy. This is a huge problem. This is what we want. This is the way it used to be. Getting there is going to be very difficult. So in the meantime, this is a possibility, using targeted removal of ant colonies in very sensitive areas. We've done some preliminary work showing that with ant baits, which you put on the mound, you can knock out a colony. There are special ant baits that have usually been used for fire ants and more pest species, but we have very limited, or very little peripheral damage, if you will, collateral damage. We can knock out ant colonies. And this might be a reasonable tactic. For example, if you have a really good year, or even not a good year, but a year where you've got plants that are growing, and it's like, you know, we're going to knock out this colony right here because they're going to eat a lot of those seeds. We want to get this slick spot refreshed with seed. Let's give it a break. And we can do that in a targeted way. That's one possibility. Something that I'm looking at right now is the introduction of seed. Introducing seeds to slick spots, either to introduce to an area that looks like that's not occupied currently but has suitable habitat, or to augment populations which are not really doing that well. But there are issues with this, and this is something I'll just mention in passing, and we can talk about later if you'd like. Well, one, you put seeds out, I've already shown you the answer to eat them. That last experiment, 3 days later, 200 seeds gone from each cage, they'll eat them. That's a short-term problem and a long-term problem, because even if you were to target and kill an ant colony now, or, well, say you did that, but more are going to move in. So you have plants that you've seeded out there, they start to grow and flower, the new ant colonies are out there, and then they eat those seeds. It's still going to cause problems for sustainability. This is an issue you need to deal with ecologically. If you're going to put seeds out in the field, well, where did you get those seeds? Are you contaminating the gene pool of the local population by taking seeds that don't belong there? We've actually looked at this in the past. We've done a genetic analysis, and in the Snake River Basin, there's a lot of uniformity in the genetics, so we're not really concerned about that. But if you're going to introduce stuff, you should know that. The garbage area is different from this area here. You wouldn't want to use garbage seeds in our area because you'd be introducing genetic variability that doesn't exist here. Even if you were to get seeds in there and plants growing, then you got to ask questions about, do you get the pollinators to mate so they can actually produce future generations? I mean, there's other problems. If it's an area where there are no pollinators, just having the plant grow isn't going to help you. So that's kind of where I'm going with things. I'll just leave you with a final experiment I started, where I'm looking at times to introduce seeds to the to slick spots. The idea being that summer introductions are when the seeds would normally drop to the ground. An idea I've been thinking of is, so here's an ant colony in the slick spot, and here's where we're releasing seeds. That's easy pickings. But what if you introduce the seeds in, say, October or November, when the ants have already gone down for the winter? It might get those seeds a chance just to escape that initial threat, so that in the spring they're buried and then they can germinate, and it might be a way of just getting past it. So I looked at summer and fall introductions, and then I also did more of that coarse versus fine soil to see if that made a difference, and ants versus no ants. And that's what's represented here by these cages. What I have is a cage that is in the simple soil, so in this flat soil in the middle. No ants can get in there. Here's on the simple soil, but it's raised so ants can get in. And I did the same thing in the coarse soil at the margin. And we did get plants to grow. About 160 plants across 9 different slick spots grew. That was adding, I think, 200 plants per cage. There were 8 cages per slick spot, so it's not a lot of plants, but seeds don't always germinate the first year, they might germinate in subsequent years. So we can go back year after year. Some germinated. This one even produced flowers and fruit. It was the only one. There were 4 that flowered. 3 of those flowering plants never got pollinated, they withered and died. There's your pollinator problem. You gotta somehow figure that out. The pollinator work we did earlier showed that they're generalists. Lots of insects will visit the flowers, but you might need to have a certain concentration of flowers to even get pollinators to come in at all. I don't know why this one worked. It might have self-pollinated. Okay, here was the effect. So summer and fall introductions. The black bars represent simple area, the flat hard pan, when ants were excluded. That's where we got the most rosettes, the most plants germinated. No ants could get at them. If ants could get out of the white bars, the simple soil with ants, we got none there. We got a couple here in the fall. Ants were finding these seeds and taking them back. And then in the coarse soils, same problem. With no ants, they actually didn't do that well. Those margins just don't appear to be that great. What was happening is I was getting all sorts of mosses and lichens and grasses growing up inside those pots. And then there is a bit of a problem here. The summer and fall didn't look that much different. The fall introduction was a problem. I would have expected that this one bar here would be higher because ants weren't there. But we put these things out in mid-October, and then it went up into the high 70s. And ants were out in quiescence. When it's cold, they stay down, then it warms up and they're out and they're out. And they were out there, I could see them eating seeds. And so really, these are kind of like the same thing. So last year I went out in November when it was really cold, put them out, and we'll see this spring if they actually do better at this time. But even so with this, it tells us a little bit about what ants are going to do if you put seeds out. You know, a lot of management decisions, it's like, oh great, we'll put seeds out and we'll replenish, and if you don't ask the right question, you're wasting resources. If you're just to throw seeds out there right now when there's ants, they're just going to eat them. And if they're hard to collect seeds and they're rare, you might not want to do it. You might want to think of different methods. Even if you get seeds germinating on a higher number, then you got to deal with the pollinator issue. And so that's where we're kind of going with this now. This coming summer, I want to look at a larger scale introduction study, again with ants, no ants, timing of adding plants, but also trying to do it in concentrations that might lead to more flowers in a locale, and seeing what it does to the pollinators, if you can actually get pollinators, because you can't really ramp up until you figure out what's going on. So, just to finish up, this stuff all has to get funded. This is one of the things we do as researchers is beg for money. We propose projects. These are the agencies that have helped me out over the years. The Idaho Army National Guard has been one of the best. They have the Orchard Training Area. It's called the Orchard Combat Training Center now. They have a lot of land to manage, and they're really serious about conservation. And they do stuff with reptiles and mammals and sagebrush and cheatgrass and lepidum. And every year they're hiring students, and I think we've got a number of faculty getting research. They've been great. And they're the ones that are going to help me with the seed introduction stuff next year. along with the funding comes a lot of undergraduate help. Sorry, Amy. Amy was one of them. She wasn't involved in the seed studies, but this is Amy back in 2002. But lots of people over the years, and there are more. There are some people that come in the lab and help count seeds, and I didn't get all their names. But help out with the research. It's a good opportunity. It's how I got started as a researcher. Helping someone do their master's, and it was sort of something I like to continue on because it was really, when I got my first job doing that, everybody else was sort of trying to figure out what they wanted to be, and I was just like, I know what I want to do now. I hated botany classes at the time. Even though I'm working on plants, but it was like, I'm not going to let botany screw up my chances of getting into grad school, so I tried really hard and did well in botany, even though I was bored by it. I had direction, I knew where I wanted to go, and it really motivated me to do that. So undergraduate experiences are really important. And then a whole list of grad students. I mean, there have been a number of them working on Ophidium. That's the clean one. But we had people initially working on pollination, genetics and population structure of the plant. Then Josh came in with seed predation. Matt did it. I didn't talk about Jen Crossman's work. This was a student who in her second year encountered the massive drought that killed all our plants. And so she's still working on her thesis. Talk about Michelle's stuff. I've got a new student coming in next fall to take over some of this work. Not going to be doing the seed counting and grains of sand stuff. If you see Michelle, know her, tell her you're sorry on my behalf. She's been a real trooper through all of that. We'll get her out of here soon. So that's all I had to do talk about. Hope it was informative, and also in the context of predation. I know it's a weird topic for predation, but you can see that the ideas extend beyond animal. Questions?
So you talked about one of the ideas or options being ant removal with pesticides, but I was wondering, I don't know that much about the ant's role in that sort of ecosystem. What is eating ants, and what are ants? Well, ants are obviously, you talked about what they're eating, but I'm just curious of what kind of their role is and if that would have huge environmental impacts in that area beyond just the plants being safe.
That's a great question. It's the same question the Fish and Wildlife Service brought up a few days ago. When I proposed doing this, they said, well, you're going to need to present to us some information about trophic levels and what this might mean. They're worried about poisoned ants poisoning higher trophic levels. So not much eats these things. Their biggest predators are horned toads, horned lizards rather. Phrysanoma is the genus. And at the site where I want to do this work, we just haven't seen any in 15 years. There are in Glens Ferry. These lizards specialize on ants, and they'll often just sit on a colony and just pick off ants. They just aren't abundant around here. Lots of other things have been associated with them. Sage grouse will eat them, but we don't have any sage grouse existing where these populations are. There are some spiders that will eat them. That's a potential problem. As far as the baits go, we use something called Amdro, which is registered for ant killing. And you put on a couple teaspoons on the ant mound, and the ants grab it and take it down into the mound, so off the surface. And they feed it to the queen, and it kills her, and then that basically neutralizes the ant colony. I've looked up some of the information about toxin levels on this. It would take about 600 doses to be an LD50 for a dog. So, I mean, that a lethal dose in 50% of the time. 600 doses, I mean, we're going to come nowhere near those levels, and it's being taken underground. And so it's considered very low toxicity, except in aquatic environments. We are in desert environments where that's not an issue. It's bad for aquatic invertebrates and things like that. So I think overall it's low risk. And also, we're not talking about, it's not like an aerial application where you're knocking out hectares or whatever. We're talking individual ant colonies targeted for removal. Now we are interested in, so you can kill an ant colony. Does that just open a space where another one just starts to move in right away? We want to know that. beyond that, I'm not too concerned. Ecologically, the ants are important, but we're not hitting them at a large scale to change that. Also, the ants they're kind of, I've often viewed them as being kind of like an invasive species within their own natural range, and that we're changing the habitat that has greatly favored the proliferation of these ants. They're native to the area, but normally they're in much lower numbers and much more patchily distributed than like I showed you on that map where they're just everywhere, because that's not how this area should even look. So the ecology of the system is entirely changed too.
So I know you said that the cheatgrass is like really hard to handle and that's why they don't need it. Do you, and also the ants, like, you know, they don't travel, what is it, like how many
About
meters
20 meters.
from there?
It's a long way actually.
Is this like kind of a stupid thing to be like, okay, let's put ants in a heavily cheatgrass area so they have
Or
no,
promote cheatgrass.
like, you know, like,
Well,
they have no choice but to eat just
Yep.
cheatgrass.
That's, yeah. It's funny, that always comes up at meetings because we sort of laugh and say, oh, what are you saying? You know, the natural grass, the Sandberg's bluegrass, they really like. And they could see that. So remember how I showed they eat bluegrass? These ants will do absolutely fine where there's no lepidium or mustard. The bluegrass is great for them. Cheatgrass is not. People are saying, so more cheatgrass with limited ants. The problem is, and I didn't get into it, is that cheatgrass has its own suite of problems. And one thing it does is invade slick spots. It actually pokes away at the margins and eventually just grows over them and they disappear. There are places now, like at that main site I work at, where I can see the remnants of slick spots that I used to work at 10 years ago. There'll be depressions and the cheatgrass will be a little purplish color. I don't know, maybe the moisture level's different, it grows a little differently, but it's totally removed the slick spot. And occasionally you'll see a little peppergrass plant growing in there, a seed that finally survived, but it's basically eliminating habitat. So while it may be good at not being food for ants, right, it's terrible for the environment overall because it eliminates suitable habitat for the plant.
So what I think I was trying to say, like, put the ants in cheatgrass, like where
Can't make the ants go anywhere.
you can't, like
No.
you can't just place them somewhere.
Nope, they're going where they want to go.
Okay.
So when the ants breed, what happens is they, a certain time of year, you get winged individuals come up,
Okay.
and then they fly to a mating site. It's usually an elevated place where they all sort of congregate, and then they disperse from there and start breeding. We can't make them go
You
anywhere.
can't make
In fact, they would go to areas that are preferable to them.
Okay.
They often go into slick spots because they're nice bare patches where they don't like vegetation around them, so they just go right in the middle of a slick spot, which is also where lepidium grows. And so there's a lot of slick spots that have ant colonies right on the margin or right smack dab in the middle of them.
What's the function of peppergrass in this ecosystem? Like, why is it so important to preserve? I guess
Yeah.
what's beyond just saving an endangered species for the sake of saving an endangered species.
Yeah, that's a good question. It's an age-old question in conservation biology is why preserve it. It doesn't have any specific value in any economic terms. It is a plant that is specialized to a local habitat condition. It has a very limited range and it exists here. And so from a conservation perspective, we are tasked with making sure that human activities aren't responsible for the elimination of a species. And our activities are causing problems. Cattle grazing is a huge problem. But, you know, so again, as my position of not being an advocate, I don't worry about that question. My question is my approach is to provide information that others can use. I think there's inherent value in conserving species, especially if our own activities are the reason it's not doing well. Would anyone really miss it if it were to go extinct? Not in any tangible way, just that.
No, Colin, because I use the PlayStation. I use the PlayStation. No.
I made dinner, by the way. It's shitty. It's shit dinner, but you're welcome.